Welcome back to part 2 of the lecture on chapter 1. We're picking up with some of the research that was done in what's referred to as the golden age of microbiology. What that means is that in the late 1800s and early 1900s, a span of about 50 years, there were huge uh, strides made in understanding, well, first of all, figuring out that microbes existed, and then secondly, what they did and how to use them or how to control them. So one of the big questions at the time, it seems silly now, but at the time it was real to people. There was this idea of spontaneous generation of life, microbial life. For example, if you bought some meat and you didn't have refrigeration, but you were trying to keep it so that you and your family could eat it later. If you didn't eat it soon enough, stuff started growing on it, even though you packed it away very carefully and you didn't see anything on the surface of this thing. Later, it spoiled. And so there was this idea that these microbes could come into being just from, thin air. People didn't understand what caused fermentation. There were, you could save some juice and I want to drink that later. But if you save it for later, sometimes it changes the characteristics of that juice and made it taste different. It wasn't as sweet as it used to be, and now it burns when you swallow it. And then on top of that, what causes disease? Why do some people get sick? And then how can we prevent infection and what we call disease? There was finally this idea that there were germs, so germination, there were things that could start life, but there was these little creatures that could create disease in people. And there were plenty of folks who had thought about it and tested it, but Louis Pasteur is the guy who's credited with figuring out a lot of this information. He was the one who showed the connection between food spoiling and the presence of microbes. And that showing that connection was also really critical in the scientists in the air at the time, kind of coming around to the idea that food spoilage is not so different from body spoilage, and that some of these diseases that we were seeing were actually the result of the action of microbes. Then, so Pasteur's famous for a lot of different things, but he's pretty much, the dude was interested in little organisms and their impact basically on industry. I mean, if you can think about the industry of food, wine, cheese, Pasteur was on it. We know the process of pasteurization mostly around milk, but it's also applied to fruit juices and apple cider and that kind of thing. All right, so Pasteur, silkworm, what the heck does silkworm have to do with milk or cheese? Silkworms make, you guessed it, silk, which is financially really of interest to people. That's why Pasteur was in on this. Anyway, the whole point of this is, if you're going to keep a whole bunch of silkworms together so you can, harvest the silk and use it to make fabrics that you can sell for a lot of money, you're going to find that sometimes some of these silkworms get sick and then it spreads through the population, which is, devastating to financial backers because they spent all this money and energy buying and building up the colonies of these things. Anyway, the point is, Pasteur identified that there were two different diseases that affected these populations of silkworms. One of them was a fungus and one of them was a protozoan. But the point is, there's a connection between microbes and disease. This was in a different species than humans, but it's not that huge a leap to make, that thinking that microbes might have an impact on human health as well. And then indirect evidence for this hypothesis that microbes were related to disease, these came in a number of different places. Semmelweis, he was a physician, and he insisted that his residents, so let's see how to put this. So there were medical students who would learn about human anatomy from dissecting cadavers. That's not hugely new information. What was new was his physicians, the people training under him. He said, you know what, before you go help with the labor and delivery and delivering babies, wash your hands after your hands have been in a cadaver. Wash them off before you go help with delivery. And amazingly, childbirth fever rates started decreasing in his hospital because he said, yeah, we're the ones who are introducing the microbes that are causing these women to get sick and die after childbirth. They were perfectly healthy, and we were doing that. All right, so that's indirect evidence. And then Lister is another physician. His use of disinfectants in management of surgical wounds. Sorry, let me try that again. Management of operations, surgeries, decreased how common it was for people to get infections of surgical wounds and die of infection after surgery. You may recognize this name from a product, an old product now called Listerine, which was used to kill off microbes. All right, and then finally, we get to the clear-headed Robert Koch. He was a natural-born scientist, and he was applying, this very just beautiful clarity of thought. I mean, like, if you're going to figure something out, what are the steps you have to follow? And Koch set out these 4 steps of this is how you figure out what microbe is causing what disease and how you do an experiment to test your idea. And it's this. So they've come to be known as Koch's postulates. And it's just here's your map for how to figure out if there's a connection between this microbe and this disease. First, the suspected microbe needs to be able to be found in all cases of the disease. Every single case of the disease, if you find this microbe, you've got a good suspect. But that same microbe has to be completely absent from all the healthy samples. So if you've got patients who have the disease and you sample sputum or whatever you're taking to look at, if that microbe is present there, but take the same kind of sample from the sputum of a healthy patient and you don't see that, okay, that's step 1. Then step 2 is get that microbe and grow it outside of the host so that it's no longer going to be transferred from one human to another human or one sheep to another sheep. You've got it in the lab or you've got it in some kind of dish outside of the host. Then this is where you get to the kind of unethical part of this. Step 3 is then that microbe has to be introduced to a so-called healthy host, an organism that hadn't previously shown signs of disease. And then if that experimental subject who's given this microbe, if that host gets the disease, that's step 3. So you've got some pretty good evidence. But Koch didn't stop there. He said, nope, you got to do one more step. And that is take a sample from that now sick host and see if you can identify that same microbe. And if you can, you have now successfully demonstrated that there is a connection between this microbe and this disease. Koch used these steps to demonstrate the connection between a specific disease himself. And the disease we're talking about was anthrax in cattle. It was also in sheep, but not in humans, but anthrax in cattle. We knew that there was anthrax. We knew that there was a disease. We didn't know what caused it. Now we've identified, he identified this bacterium, and now it's been given the name Bacillus anthracis. That's the microbe that causes anthrax. Ta-da! These very same steps were used in figuring out what it is that causes ulcers in humans. It's a great story, and if you're really interested in it, I can tell you about it some more, but let me know that you're interested. But yeah, it's a fantastic story, and I'm not recommending that you follow these steps yourself because it was some scientist who experimented on themselves. I shouldn't laugh, but it's a great story. All right, so that is the end of the slides I have for you. What we have for you to do this week is read through chapter 1, excuse me, in our textbook. And there's a lot more information in the chapter than I can cover in a lecture. But the materials that I touched on, I would recommend that you read that in a little bit more detail. And there's a homework assignment in mastering. It will be linked in Canvas, and I would like you to do this first homework assignment in mastering. Oh, this will be the second homework assignment. The first homework assignment in mastering is just learning how to use mastering and figuring out how to drag and drop things. If you have any questions, let me know. Send me a message, and I'll talk to you later. Bye.